So this is chapter 4, and it's about the anatomy, mainly of prokaryotic and some eukaryotic cells. So we have gone through this before, but just to brush off cobwebs, the prokaryotic before the nucleus, and that's going to be the bacteria. Every other cell on the planet is eukaryotic. So this means it has a true nucleus, and all of our mammalian cells are eukaryotic cells. And this is just going into a laundry list. Here you have one circular chromosome, here you have paired chromosomes. So the thing that this doesn't get into, but I'm going to mention, is that prokaryotic cells do not do sex. There's no sexual replication. There's no eggs. There's no sperm. That came later. That is actually an evolutionary advance. The eukaryotic cells do sex, and they got complicated very, very quickly, and that's fantastic, except that with sex came death. Theoretically, prokaryotic bacteria are immortal. They can be killed by obviously different things, but as far as their division, they clone themselves, they make copies of themselves. So we don't. I am a unique blend of my mother and my father. I was born, I have a a lifespan, and then I will die. And that is the fate of all eukaryotic cells. But it does allow you amplification of your chromosomes. You can do division of labor. You can make tissues. You can make organs. You can make organ systems, and you can get big. So that's kind of the plus side. Death is kind of the minus side. We have 46 chromosomes, 23 from mom, 23 from dad. So we are diploid, and we can have things that are dominant or recessive, and bacteria not so much. Histones are a way of taking very large chromosomes and condensing them. Since the bacteria only have one, they don't need histones. They also don't have organelles. We have lots of organelles: mitochondria, lysosomes, endoplasmic reticulum, nucleus, etc. Now, prokaryotic bacteria have cell walls, and we'll get into that in a couple of slides. And peptidoglycan is the kind of the glue and what's in common in the cell wall of all bacteria. The plants on the planet do have cell walls. We, however, do not. And then division is binary fission. You get big, make a copy of your one chromosome, divide in half. There you are. And it's very much cloning, so that you get the same cell when you divide. Now, when you make eggs and sperm, that's quite different. So the other cells in your body, besides the sex cells, the gametes, are the regular body cells, and they do mitosis. So when they do mitosis, there is something called a mitotic spindle. And we're not going to get into mitosis, but just to mention that. The size, again, they're about 10 to 100 times smaller than our cells. And I don't want you to memorize that they range from 0.2 to 1 millimeter. But these are the basic shapes. You have cocci, which are round. You have bacillus, which are rod shaped. And then you have spirillum that are spiral shaped. They also can be arranged differently. They can be single, or some of them can be arranged in two. That's diploid. You can also have them be clusters, like staph. You can also have them be chains, and chains of cocci are called streptococci, and chains of bacillus are called streptobacillus. Now, there are some bacteria that do need an aid to move. You know, if you are a rod shape, it's going to be harder to move you than if you are round. So these rod shaped bacteria or the spiral-shaped bacteria will have a flagella, and that's an organ of locomotion that is going to spin and is going to propel the bacteria because they just have too much drag. If you're a ball, like a coccyx, you can just roll. But if you are a bacillus, if you are a spirillum, then in order to compete, you need an additional flagella in order to keep up with those cocci. And this is just showing you the flagella, which is made up of a protein, actually hooks into the cell wall and plasma membrane, and that way that actually generates, it's almost like a motor to get that flagella spinning so that the cell can move. And here we go. So here we have flagella. They can be one, they can be two, there can be many, they can be on one side. And I am not going to test you on monotrichus, amphitrichus, lophotrichus, or peritrichus, but just to show you that depending on the challenges, does the bacteria have to swim through mucus? Well, it'll be better if you are peritrichus. If you are just talking about something that is more water-like, then you probably be fine with monotrichus. But the flagella definitely do have to be there to help that bacillus to move. And now what we're going to get into is the cell wall. The cell wall is that outer, mainly protein, protective layer, because the bacteria don't have a lot of control about their environment. They could be in your body in 1 minute and be flushed down the toilet in the next, and vastly different environments, and the cell wall is going to help them survive that. All right, so peptidoglycan, here we go. I liken the peptidoglycan layer to a barrel, like a barrel being very protective, covering over the bacteria. And like a barrel, you're going to have horizontal and vertical pieces of wood. Over here, you're going to have horizontal and vertical molecules that will make this and kind of weave the peptidoglycan cell wall around the cell. The word peptide is a small protein. The word glycan refers to sugar. So what we have is we have what I would call the horizontal part of the peptidoglycan cell wall is actually made up of sugar subunits. And then the vertical that is going to link everybody together, in this case is that blue and green, those are the peptides. So the way that is structured is more like weaving a cloth. So you have the sugar units and they are stacked one on the top of the other, but you're going to weave the peptide chains so that you really create a very solid, strong structure. And Christian Graham, who was a Danish bacteriologist way back in the year of the flood, had a staining procedure, and he started to realize that there are two groups of bacteria, and it was named after him. And so we have gram-positive and we have gram-negative bacteria. This was back in the 1900s, and biochemistry was not a science yet. And what he didn't realize is that gram-positive and gram-negative bacteria are actually named because of differences in their cell wall. So both cell walls of gram-positive and gram-negative do peptidoglycan. That's it's like you got to have that barrel. So you have a peptidoglycan here for gram-positive, and you have a peptidoglycan layer here for gram-negative. Bacteria are very wily but intelligent cells in that they have divided your body in half, so that from your waist on up is the where you have gram-positive, and the waist on down is where you have gram-negative. That way they're not competing with each other, but it also means they have different challenges for survival. You have mainly in the respiratory tract gram-positive organisms. So pneumococcus pneumonia is gram-positive, um, and so forth and so on. So most of the organisms that can cause respiratory infections tend to be gram-positive. Streptococcus pyogenes, which causes septic sore throat, is gram-positive. So the strategy is that when you're breathing in and out, there are a lot of pressure changes, and you want, therefore, to have a very strong, um, almost like an armor-like cell wall. So gram-positive cell walls have an additional layer that kind of tightens the weave, and it's called teichoic acid. And these teichoic acid chains fill in the spaces and makes the cell wall even stronger. In contrast, if you're gram-negative, you are your diarrheal diseases, you are your dysentery diseases, these horrible diarrheal type of diseases. So you're the Salmonella, you're the Shigella, you're the Proteus, and these are all gram-negative pathogens. Now, their problem is not the pressure changes. Their problem are all the chemicals that you're being, because you're going to eat, let's say, Aunt Mildred's chicken salad that has been out in the picnic table all day long, and you're going to look forward to salmonella poisoning that evening. And in that particular case, it's all of your digestive enzymes. It's your digestive enzymes, it's the hydrochloric acid in your stomach. So they have to be chemically strong. They don't have to be physically strong. So one of the big things that's very difficult for our body to break down is fat. So the idea is that you're going to layer on 2 fat layers on top of the peptidoglycan layer in order to get from the mouth to the esophagus, to the stomach, to the small intestine, until finally you're in the large intestine. And that's when you're able to colonize and cause disease. So you're going to have 2 fat layers. You're going to have this, it's called a lipoprotein layer, or they're calling it a phospholipid layer. And you're going to have I'm sorry, here's lipoprotein, there it is. And then the outer layer is called lipopolysaccharide. So the lipoprotein layer, here's your peptidoglycan. So you have the peptidoglycan structural layer that's protecting everybody. Then your first fat layer that goes around is going to be your lipoprotein layer. Then the second layer that is put down is going to be your lipopolysaccharide layer. So between the 2 fat layers, you survive. You don't have to worry about pressure. And the reason why you don't have gram-negatives in the respiratory tract is that lipids kind of like butter, and it would just smish under high pressure. And on the other hand, if you had a gram-positive, you don't have gram-positives causing dysentery. And that has to do with the fact that they're mainly made up of protein. So as soon as they hit that stomach, they're going to be disintegrated and destroyed. So it's interesting how they don't want to compete with each other, but they had to make modifications of the cell wall in order to thrive in the environment that they chose. Okay, so this is showing you both of them are going to have the peptidoglycan layer, one thick, one thin. The teichoic acids is going to make it tighter and more protective. Now, the acid fast cells are just the cells that cause TB, tuberculosis, so I'm not going to emphasize that, and it's not going to be on the exam. Gram-negatives, no teichoic acid, and they really should have listed the 2 fat layers. Here, what should be is, and besides an outer membrane, what they're really referring to is that lipoprotein, lipopolysaccharide layer, and that is what gives them chemical resistance. Okay, so this is extra material. I am not going to hold you responsible for it, but it has a nice diagram. And this now then gets into lipopolysaccharides of gram-negative. Right now, I'm going to have a separate unit on toxins and antigens and phagocytes and complement and antibiotics. So for now, I'm going to skip that, and that is not going to be on the Moodle quiz. And this is just another lovely diagram of the gram-negative. Now, the last but not least organelle that I'm going to be talking about in this particular chapter is the plasma membrane. Bacteria invented them and passed them along. Every cell on the planet has a plasma membrane. It's going to be a membrane that will allow discernment, selectivity of what goes in and out of cells. So it's composed of a phospholipid bilayer with specific proteins that act as receptors. So when you took A&P, you learned a lot about plasma membranes, and they are exactly the same in bacteria as they are in us. So that is the end of this particular chapter, and I will see you soon for the next one.